Good morning, students. I'm your regular anchor on this course in Jeddah. This is our second topic or second lecture we're about to have now. And I will briefly let you know what we're going to be discussing. Previously, we talked about classification of carbohydrate definition, where we talked about monosaccharide, oligosaccharide, and polysaccharide. And in this second week, I want us to revise what we have done, more especially under carbohydrate metabolism, where we will be talking about digestion, absorption, transport in tissues, and also glycolysis. Under that, we are going to be talking about glycogenesis, glycogenolysis, and gluconeogenesis. We will also discuss tricarboxylic acid cycle and hexomonophosphate shunt. These are the topics we are going to discuss as we move along. And I will surely provide the materials for you so that you go through before our next class. Thank you. Carbohydrate metabolism. The carbohydrates are source of energy for animal nutrition. The monosaccharides and oligosaccharides are efficiently metabolized by simple stomach animals. On the other hand, ruminants contain microbes which secrete enzymes capable of degrading cellulose. Glycogen is a polysaccharide found in animal and fungal cells. Glycogen is a storage form of carbohydrate and is readily utilized when there is deficiency. Objectives of this topic. At the end of this unit, you should be able to understand the metabolism of carbohydrates in terms of their digestion, absorption, and transport in the tissues. Know beyond reasonable doubt the metabolic pathways of carbohydrate use and storage consist of glycogenesis, glycogenolysis, glycolysis, and hexomonophosphate shunt, the citric acid cycle, and gluconeogenesis. Digestion. The dietary carbohydrates that are most important nutritionally are polysaccharides and disaccharides. Since free monosaccharides are not commonly present in the diet in significant quantities. There is, however, some free glucose and fructose in honey, in certain fruits, and in the carbohydrates that are added to processed foods. The cellular use of carbohydrates depends on their absorption from the gastrointestinal tract in the bloodstream, a process normally restricted to monosaccharides. Therefore, polysaccharide and disaccharide must be hydrolyzed to their constituent monosaccharide units before utilization. The hydrolytic enzymes involved are collectively called glycosidases, or alternatively carbohydrates. Absorption and transport. The wall of the small intestine is comprised of absorptive mucosal cells and mucus-secreting goblet cells that line projections called villi that extend into the lumen. The absorptive cells have a hairy projection-like surface on the lumen side called microvilli or brush border. A square millimeter of cell surface is believed to have as many as 2 × 10⁵ microvilli projections. The anatomic advantage of the microvilli structure as it relates to the absorption of nutrients is that it presents an enormous surface area to the intestinal. It has been estimated that the absorptive capacity of the human intestine amounts to about 5,400 grams per day for glucose and 4,800 grams per day for fructose, a capacity that is, of course, never challenged in a normal diet. Glucose and galactose are absorbed across the gut wall by active transport, whereas fructose moves across by facilitated diffusion. Active transport implies that the process is energy requiring and that a specific receptor is involved. The exact nature of glucose and galactose carrier is unclear, but is known to be protein complex connected to the sodium and potassium ion, that is ATPase pump, adenosine triphosphatase pump. Glucose appears to exit the mucosal cell by different routes. One, approximately 15%, leaks back across the brush border into the intestinal lumen. About 25% diffuses through the basolateral membrane, but the major portion, approximately 60%, lives via a carrier in the serosal membrane. If mucosal cells are poisoned with chemical blockers of oxidative phosphorylation, the transport of glucose ceases. The fact that some cases of glucose malabsorption have been attributed to reduced numbers of specific carriers testify to the importance of this metabolism. Following transport across the gut wall, the monosaccharides enter the portal circulation and distribute among various tissues in the body. Galactose and fructose are readily taken up by liver cells via specific hepatocyte receptors and are subsequently metabolized. The metabolic fate of the monosaccharides depends to a great extent on the energy demands of the body. According to these demands, the activity of certain metabolic pathways may be stimulated while others may be repressed. The major mediators of this regulation are hormones such as insulin, glucagon, and the glucocorticoids, which activate or inhibit specific enzymes within the pathways, and allosteric enzymes, which are stimulated or repressed by certain compounds formed within the pathway in which the enzyme. An allosteric or regulatory enzyme is said to be positively or negatively modulated by a substance modulator according to whether the effect is stimulatory or repressive, respectively. ATP, adenosine triphosphate, and its dephosphorylated products, adenosine monophosphate, formed from ADP, adenosine diphosphate, by adenylate kinase, as shown in the reaction, can modulate certain allosteric enzymes through opposing effects. This exemplifies the link between energy and the demand and allosteric enzyme modulation. As ATP accumulates, for example, during a period of muscular rest, it can negatively modulate certain regulatory enzymes in the energy-producing pathways so as to reduce the production of additional ATP. An increase in AMP concentration conversely signifies a depletion of ATP and the need to produce more of this energy source. In such a case, AMP can act as positive modulator of enzymes. The enzyme phosphofructokinase, which catalyzes a reaction in the glycolytic pathway, is modulated by both ATP and AMP in the manner described. The ratio of nicotinamide adenine dinucleotide hydrogenase to nicotinamide adenine dinucleotide, it also has an important regulatory effect. Certain allosteric enzymes, for example, are responsive for an increased level of NADH, which therefore regulates its formation through negation. The metabolic pathways of carbohydrates use the storage consists of glycogenesis, glycogenolysis, glycolysis, and hexose monophosphate shunt, the citric acid cycle, and glucolysis. Here, the diagram is showing us the different pathways of this carbohydrate metabolism, which we have just mentioned. That signals the end of glycolysis. But in the other way around, we said glycolysis is not, pyruvic acid is not the end product of glycolysis in the presence of oxygen. In our next class, we will observe how the TCA cycle, tricarboxylic acid cycle, or the Krebs cycle, sets in. It comes in if there is presence of oxygen. The pyruvic acid or pyruvate will now be converted to acetyl-CoA. In the presence of this enzyme, pyruvate dehydrogenase complex, and the whole thing undergoes oxidative deletion. So at the end of it, at this TCA, other carbohydrate metabolism, which involves the PPP, the glycogenesis, gluconeogenesis, and glycogenolysis. As you go through my lecture slides.